Okay, chapter 7, section 14, attenuation. Attenuation is a mechanism for controlling, well, production. Production controlling at this, though at the transcription level event. You are using translation as well as transcription mechanisms to control yes, no, whether or not transcription can occur. It's going to be based on what is referred to as a leader sequence. It's the first part of the messenger RNA the ribosome will run into. It's part of the genetic structure of an operon. It's up there with the operator and the promoter sequences. This leader sequence is going to control yes, no, whether transcription can occur. No transcription, no translation. But you have to remember that the transcription has to begin and translation has to begin before the leader sequence can be expressed. We're going to be using the tryptophan operon in E. coli as our example here. It should be, noted without stating it, but I'm going to state it anyways. But here, since we're talking an operon, and we're talking about transcription and translation occurring at the same time as part of the same event, that this is going to be a prokaryotic event only. Reason being, in us eukaryotes, transcription and translation seem to be separated by that pesky nuclear envelope. Not going to have that nuclear envelope in a prokaryotic cell. So what you see here is promoter, operator sequence, the L, the leader sequence, and then the structural genes for the tryptophan operon. These are all going to be genes responsible for the uptake and the certain substrates, and then a modification of those substrates to form tryptophan. Here's the leader sequence, and notice this tryptophan. This is something you'll see throughout numerous different operons that will utilize this whole leader sequence thing. It's a conserved, pattern. So this leader sequence is going to make a small polypeptide that can, at high concentrations, high levels, interfere with the transcription of the operon itself. What we see happens, like I've stated 2 slides ago, transcription and translation. We always, when we're lecturing and we're showing pictures of it, we always portray them and discuss them individually, which unfortunately gives many students this mindset that in the prokaryotic cell, that these are happening in 2 different places independent of each other. And that's furthest from the case. When the RNA polymerase hops on the double helix of DNA and starts going down it, making messenger RNA, as soon as that messenger RNA gets long enough, a ribosome will hop on. And that messenger RNA gets longer and longer. There's one ribosome, there's another, there's another, there's another. And the whole time the RNA polymerase is just marching down the gene or the operon. So you actually get transcription, translation occurring at the same time. Just transcription's got to go first to start things, and then quickly right after that comes translation. You have to take into account those stem loops we've talked about over the past 2 sections. Those stem loops that give the RNA, 2-dimensional RNA strand gives it 3 dimensions. These stem loops are going to inhibit movement of the ribosome down the messenger RNA, and it's also going to inhibit, remember we talked last section, in section 13, the movement of RNA polymerase. Exactly how that does it, don't know, don't care, don't worry, just that it does. So what we see happens with the tryptophan operon is that leader sequence, if the leader sequence, you see it going, and if there's plenty of TRP, tryptophan, the sequence is going to form stem loops, okay? Going to happen anyways, but in the presence, high level, low level of tryptophan, it forms different patterns of stem loops. If there is little tryptophan, the ribosome pauses, and the stem loop pattern will be such that it pauses, but it will then continue on later because, tRNA, tryptophan tRNAs will remove what little tryptophan is there, causing a change in the stem loop pattern. But if there's too much of it, too high a levels, yes, it's going to form a specific pattern of that growing messenger RNA. That growing pattern of stem loops is going to cause the ribosomes to pause, and it's also going to interfere with movement of the RNA polymerase. Interfere with the movement of RNA polymerase falls off. The ribosomes pause too long, they also dissociate. So high levels means there's no reason to make more transcribe the operon, there's no reason to make more tryptophan. Whereas if tryptophan gets low, everything pauses, but the level of tryptophan keeps decreasing, and then those stem loops resolve, transcription, the RNA polymerase starts back up again, and then the ribosomes start back up again. So you see here the comparison of the two. Excess tryptophan, transcription will be terminated because this stem loop pattern, as you see here, sequence 3 to sequence 4 causes a stem loop pattern that makes the ribosome pause, but then it also interferes with the movement of RNA polymerase. RNA polymerase dissociates, no transcription. Given enough time, this also will dissociate, come apart, and then you get also no proteins being made. And it's all because of this leader sequence here. Yet causing binding and all sorts of things in here. If there's a limited amount, tryptophan levels are low, below a threshold. Well, here's the pattern. Sequence 2 now bound to 3. Polymerase will pause, but then it'll continue on. Once it gets, enough of the stem loops rearranging, ribosome continues on, making more. But it's this leader peptide here. It's being made, causes this to pause long enough for this to form. And whether or not there's enough here causing it to pause, and where and when and how, you either get this or you get this. If you get this pattern, everything stops. You get this pattern, things pause, but then we'll continue on.